Let us talk about another important operon, which is tryptophan operon. The trp operon consists of a promoter, an operator, a leader or trpL, an attenuator, and 5 structural genes designated as trpE, trpD, trpC, trpB, and trpA. So here is the structure of this tryptophan operon. These trpE, D, C, B, and A, and this one is the trp leader sequence or trpL, and this one is trp attenuator, this is trpP, and this is trpO, means promoter and operator. So this is the sequence of also this one is the regulatory portion, and it regulate the this operon, and this is enzyme production region. Consistent with the fact that the trp operon specifies biosynthetic rather than degradative enzymes, it doesn't have a CAMP CRP activation साइट. तो जैसे आपने पिछली एग्जांपल्स देखी गैल ओपेरॉन की, लैक ओपेरॉन की, ये दोनों जो थे ये डिग्रेडेटिव ऑपरेंट्स थे, यानी कि ये डिग्रेडेटिव एंजाइम्स बनाते थे, और ये जो ट्रिप्टोफैन ओपेरॉन है ये डिग्रेडेटिव नहीं है, बल्कि ये जो है ये सिंथेसिस करता है. The operator is part of a coarse on-off control, and the leader and attenuator allow for finer control. तो दो किस्म के कंट्रोल हैं, coarse control और finer control. The leader sequence, which codes for a short peptide, is the first region of the trp operon to be transcribed. RNA polymerase then moves forward to transcribe the 5 structural regions in the order trpE, trpD, trpC, trpB, and finally trpA to form the polycistronic trp mRNA. The 5 polypeptides specified by the structural genes form the 3 enzymes that are essential for tryptophane synthesis. Early studies revealed that the trp system is turned off when tryptophan is added to an E. coli culture, E. coli growing culture, suggesting that the operon is repressed rather than induced. Repression, like induction, involves negative regulation of transcription initiation by a regulatory protein. However, the regulatory protein doesn't bind to the operator until after forming a complex with tryptophan. The structural gene for the trp regulatory protein or trpR is located at a considerable distance away from the trp operon. तो TRP रेगुलेटरी प्रोटीन TRpR और TRP ओपेरॉन के दरमियान में काफी डिस्टेंस. This distance doesn't present a problem because the regulatory protein diffuses throughout the cell. The biologically active form of the regulatory protein is a homodimer. Each subunit contains a helix-turn-helix motif that binds to a trp operator half site. The trp operator and promoter regions have significant overlap. Therefore, binding of the trp tryptophan complex and RNA polymerase are mutually exclusive. The trpR tryptophan complex functions as a coarse on-off switch that turns the tryptophan operon off when tryptophan levels are high. तो जब ट्रिप्टोफैन लेवल हाई होंगे तो ये जो है ये ट्रिप्टोफैन ऑपेरॉन को ऑफ कर देगा. अ फाइन कंट्रोल mechanism also exists that allows cells to regulate their tryptophan enzyme concentration according to the tryptophan concentration. तो tryptophan concentration को देखते हुए tryptophan enzyme concentration को control किया जा सकता है. An important clue to the existence of this fine control mechanism came from an experiment performed by Charles Jafonski in 1972, which showed that E. coli mutants that lack a functional TRP protein increase TRP operon transcription after being starved for tryptophan. If trpR tryptophan complex were the only regulatory factor, then transcription of the TRP operon should not have increased. Further studies revealed that trpE mRNA has a 162 nucleotide sequence before the first codon in trpE, designated as the leader or trpL, which plays an essential role in the fine control mechanism. Constitutive mutants exhibit a sixfold increase in tryptophan enzyme synthesis when bases 123 to 150 within the leader are deleted. तो अगर ये पोर्शन जो है 123 से 125 तक डिलीट कर दिया जाए तो 6-fold इंक्रीज हो जाता है ट्रिप्टोफैन एंजाइम सिंथेसिस. This 28 base sequence is called the attenuator. The attenuator can fold into a stem and loop structure with the potential to function as a RHO independent transcription terminator. Deleting the attenuator removes the transcription termination site, allowing RNA polymerase to complete trpE mRNA synthesis.